Good morning, guys, of grade 9. We're still talking about section 2 of chapter 6, how DNA works. So last time we started talking about the DNA, or in the previous section we know the structure of the DNA, but the question is still how the DNA works. And we said actually DNA to work, it must be translated into protein, because we know that traits or different biological processes that happen inside the cell, and even the different reactions that happen inside the cell, it needs proteins, because protein they consider as triggers for many processes and the very many important reactions that happen inside the cell. So that means DNA, as we said, is just trapped inside the nucleus and is not able to go out to ribosomes, the sites of protein production. And ribosomes, unfortunately, also they cannot pass inside the nucleus to reach DNA. So how could the code of the DNA be translated into protein if they could not reach each other? So here we said we should ask for a help, that the help of the RNA. We said that RNA is another kind of nucleic acid that look like DNA with a little differences. That RNA has a it's ribonucleic acid. It has a different nitrogen base, the U instead of T. That means contain uracil instead of titanium DNA. So RNA actually will offer a great help, okay, that it will carry the message or will carry the code, okay, or taking a copy of the code of the DNA and pass out through the nuclear membrane to reach the ribosomes. So here, let's see how the process happen or how does it work. So let's find here, guys. So the making of protein, the first step is making a protein is to copy one side of the segment of DNA. That means taking a code of one side. Why just one side? Because the two sides are complementary, so they are going to represent the same info. So it's not just it's not kind of wasting energy and time to double or to copy the two sides. Beside that, as we know that RNA just a single strand, okay, so just could be complementary to one side only of the DNA. So here like this, that we're gonna copy one side of the DNA, just the part which we need to translate, that means a certain gene or specific gene which are responsible for a particular trait. So after making this RNA, we are gonna call it mRNA. M stands for messenger, okay, because it carry a message, carry or could deliver just the code from the DNA to the ribosomes. Second step, at ribosomes, the mRNA will reach ribosomes, and ribosomes will attach with RNA. And we're gonna here ask for another help of another kind of RNA, we call it tRNA. T refers to transfer RNA. This transfer RNA will use the code, okay, which is already found on the mRNA to attach different amino acids, the building unit of protein, in their position, okay, to get the right arrangement of amino acid sequence to build the right protein. So how does it happen? Let's see here, guys. So let's move step by step. That's, as we can see, that's the nuclear membrane in purple color, and that contain holes or pores, okay, inside. So first step, that the RNA or messenger RNA will be okay copied from one segment of the DNA. So they will have a long sequence of mRNA, or that the mRNA strand. It will pass out through the nuclear membrane because it's just one strand and too small, so it could pass easily. It will pass and flow in cytoplasm. So the first step, making mRNA, it happens inside the nucleus. Then the second step, building the protein, it will happen in ribosomes. So mRNA will pass to the ribosomes and keep moving, as we can see. And here we could find that represent the different phases, okay, these different colors, okay. So we're gonna find each three phases together, we call them codon, and that's codon represent or codes for a certain amino acid, okay. So like here we're gonna find, okay, that's the mRNA could carry many codons. Each codons, okay, represent, or each codon represents for a certain amino acid. So after this mRNA passed in cytoplasm, it will reach the ribosomes, this purple shape that is the ribosome. So what would happen in that case? Actually, the mRNA will attach in the ribosome, as we can see, and once it attaches in ribosome, the ribosome will start the process of translation. It will translate these different codes into certain amino acids. So in that case here, for example, we have a lot of amino acids. This green parts, they are the tRNA, and that is different amino acid, as we can see. So for example, this tRNA, this one, where it could fit, where the shape that could fit with this one. Actually, this one could fit in that part, okay, as we can see. This one could fit here, and that one could fit here, and the part fit here. So like this, we find the tRNA, it has anticodon that could fit with the codon, okay. So regarding to this, the ribosomes, okay, inside ribosome, each tRNA will carry a specific amino acid, and it will put its amino acid in its position. How know its position? Because the tRNA anticodon will fit with the codon, so that will be the only possible place that will be placed in. So that will be put amino acid after the other, so the amino acids will attach to each other, forming a long sequence of amino acids, which we call it the polypeptide chain, or the protein, which is a sequence of amino acids. So like that, okay, that the brilliant technique of how the ribosomes will translate that sequence of nucleotides, or that sequence of codons, okay, to a certain amino acids which build a certain protein. Once protein is formed like this, that could start a certain chemical reaction or start a certain process inside the cell, and that of course will cause a certain trait to appear inside the cell or inside the entire living organism. Here then, we ask a question: could any change happen in that codons, or maybe a mistake that happened in that codons? Yes, sometimes there is a kind of a change that happened in this codon, so we call this a change in genes, or in another word we call it mutation. Any change in genes, okay, or that code, once it's changed, we call this a mutation. So we simply could say mutation is any change in the amount or the sequence of the nucleotides. So once the amount of DNA or change of positions of some nucleotides in the DNA changes, we call that a mutation. So here like this, we see that mutation change in the number, type, or order of bases on the piece of DNA. So these mutations, is it good, bad, or not, whatever, okay? So we see that actually mutation, I just have one of three possibilities, okay? Sometimes a mutation happen, but this part of DNA is not important, or this part of DNA is junk. So in that case, it will not affect anything. So in that case, we call it a silent mutation, or this mutation will cause no changes, okay? Or sometimes, okay, this mutation happen in a functional part of the DNA, and in that case, we have two options. Maybe this change is harmful, and that's actually represent 90% of mutations that happen in living organisms. Most mutations are undesirable, or they are not good, okay, they cause different syndromes and defects in the living organism. And some case, okay, the mutation, or very few cases, okay, mutations could be good or improve some traits. Like sometimes in plants, mutation may cause plants to be to form larger fruits, or sugary fruits, more sugary fruits, seedless fruits. Okay, so they first could serve better, okay, traits than usual. So that's here the possibility that happen when a mutation could happen. Okay, so we ask how mutation happen, or what causes mutation. Actually, there is no significant, okay, reason for a mutation. Mutation could happen regularly, phase, or actually they doesn't happen, okay, or they happen in random errors in the DNA while it's copies. That's mean there is no significant reason. It's just a mistake happened during the DNA replication. But there are some factors that may change those, or increase the chance of these mistakes. We call them mutagenes. So what are mutagenes? They are the factors which could cause, okay, the DNA to have these mistakes during the replication, or that cause mutation. So what are mutagenes? We classify mutagenes into two types: physical mutagenes and chemical mutagenes. Actually, physical mutagenes, they are different physical factors that could affect the body to make a mistake during the replication. For example, all kind of radiation which we expose to it every day: the ultraviolet radiation, X-ray, okay, microwave radiations, the mobile radiations, the satellites, all the radiations, okay, all these radiations actually they may cause a mistake, okay, during the DNA replication, and that leads to a mutation. Or chemical mutagenes, like different chemical substances like drugs, alcohol, smoking, okay, all these different chemical substances which pass to your cells, that also may cause a mutation. So like here, guys, we know how the protein is formed, okay, and we know if there is a mistake that could happen, and we call this mutation. So next time, inshallah, we're gonna know what are different types of mutation, or taking example for mutation, and know how, after we know the structure of the DNA and how it works, how that could help science to improve their work and so on. So study this part very well from this book, guys, and if you have any question, you can ask anytime. Thank you and goodbye.